Okay, can everyone see my screen?
Yes, we can.
Okay. Okay, well then, shall we go ahead?
Yes, please.
Okay. Okay, so my name is Tanya. I am a postdoc at the physiology department at Stellenbosch University. So Lisa asked me to share a bit about transfections. I've been doing transfections for a number of years, and I've dabbled in quite a few different methods, done transient and stable transfections. And I have had my few frustrations when it comes to optimizing it. So through that, I have learned quite a lot. So just to get us off started, transfections is basically the delivery of purified nucleic acids into cells. So most commonly, this can either be in the form of a plasmid DNA or in RNA molecules. So with either of these, you can achieve gene silencing or overexpression. But then with plasmid DNA, you can also achieve fluorescent labeling. So you can add a fluorescent marker, for instance, to your plasmid, or you can have it tagged to your gene of interest, to the coding sequence, and then you can actually map inside the cell where your protein is being localized. You can also label specific cellular compartments or just label the entire cell if you'd like. For RNA molecules, this is mostly used for if you are doing gene silencing, so that you can, and this is used with siRNA molecules, which then harnesses a process called RNA interference, which leads to specific gene silencing. So there are actually a lot of different ways to get DNA or RNA into a cell, but the most common ways are the ones here at the bottom. Electroporation is a process where electrical pulse or current creates little gaps inside the cell membrane, which then allows your DNA or RNA to enter the cell. But this requires a special machine, so it's actually not that often used unless you have a facility where you can do this. Viruses are also used quite often because they can easily bind to a cell and then deliver nucleic acid into the cell. But the most commonly used method is actually a liposuction method, where you create a complex with a lipid-based reagent and your DNA, and then that delivers the DNA into the cell via endosomes. So when you decide that you want to do transfections, and when you're planning your experiments and how everything's going to work, you need to decide what type of transfection you're going to do, whether it's going to be transient or stable transfections. Mostly people do transient transfections. Now, I've included a little basic scheme of what this would look like if you're doing a lipid-based transfection reagent. And as you can see, it's fairly simple. You just take your DNA or RNA, mix it with the reagent, incubate it, and then add it to the cells. And then normally 48 hours, you're ready for your experiment. So this is fairly simple. There's no special selection that takes place. And this is suitable for various experiments. And this is why it is the most commonly used method. But this only allows for short-term transfection. So your cells will only be transfected for a few days. If you need something that is a little bit more long-term, then you might have to consider doing stable transfections. So in this case, your cells will stay transfected a little bit longer, often for several passages. But of course, this is a little bit more time consuming as it requires selection. But if you have experiments where, say for instance, your experiment covers several days and you need transfection to stay constant during the whole experiment, then this might be more suitable. Also, if, for instance, you're going to need a large number of cells and you need it quite often, then you're going through the trouble of creating a stable cell line might actually be worth it. So normally this would be achieved with using viruses, as this process is a little bit more easier with viruses. But you can also do this with plasmid DNA and then basically follow the same process with transient, but then after transfection, you skip right through to selection. And this you'd normally do with an antibiotic, which then kills all your non-transfected cells, leaving only the transfection cells or transfected cells. And then those cells you grow up and culture and do your experiments with. So as with any other experiment, you also need to have the proper controls for doing transfections. The most important is probably your negative control. This is also your comparative control, so meaning the group with which you'll be doing comparisons. If you're doing plasmid-based transfections, this will most likely be an empty plasmid. So say, for instance, you have a plasmid and you have a sequence in order to do protein overexpression. So your negative control will then be the exact same plasmid, just without that sequence. If you're doing siRNA, then your negative control will be a separate siRNA called a non-silencing or non-targeting siRNA, which basically targets nothing in the cell. So your cells go through the exact same process, but nothing is silenced. Another important control is a positive control, where you show that the expected result is taking place. For transfections, this mostly involves showing successful transfection. So if you're doing overexpression, doing a Western blot and showing there's increased proteins is pretty standard. Same with doing knockdown, for instance, like they showed here, you can see there's less protein of this AKT1 following the knockdown. If your plasmid has a GFP or a fluorescent marker, then you can also use that to show that you have successful transfection. You can just take an image of the cells and then show that most of your cells are being transfected. And from what I've seen and from what I've seen from publications and also guidelines for publications, you will most likely have to show this when you publish. Most people expect that you need to show that your transfections are taking place as you would expect them to be. And then lastly, something that you might not see in literature that often, but something that can be quite handy during optimization is untransfected and mock controls. So untransfected is basically just what it says, cells that haven't been transfected. And then you have mock controls, which are cells where you've added your transfection reagent, but with no plasmid, or then no siRNA molecules. This helps to show you whether your cells are being affected by the process of transfection. So say, for instance, you are starting out with a new transfection reagent, or you're switching over to a different cell line, then you're seeing that sometimes the cells maybe look a bit stressed, or you don't know what's going on with them. Then including these controls will show you whether your transfection process is actually a bit too harsh on them, and then whether this is, by including a mock control, you can see then whether this is due to the transfection reagent itself, or whether the thing that you're transfecting, for instance, the protein overexpression or something, is actually causing something else in the cell. So this can actually help to help you out doing troubleshooting as well. So throughout my years of experience, I think there are three things that for me I've found to be the most important when doing transfections. Firstly, it is always optimize your transfection. And always make sure that it works exactly the same time every time. And for me, the easiest way to visualize your transfection efficiency and to make sure whether your transfection is working is by using a plasmid that also has a fluorescent marker in. Say, for instance, you want to transfect your cells that has a sequence for overexpression of protein of interest. So if you had to transfect your cells, get the protein, do a Western blot every single time, it's going to take a lot of your time. Rather, if you can transfect something that also has a fluorescent marker, after the transfection, you can quickly fix them, stain them with something like Hush or Dappy, and then you can see how many cells you have, and then how many of them is actually also expressing your plasmid. So this makes transfection a lot easier and a lot simpler to optimize. And then also, you can use this image as your positive control to show that everything's working as it should. The other thing is, as simple as it sounds, healthier cells will always give you better transfection. If your cells are stressed, they're not going to like the extra presence of DNA or the transfection reagent. And so you're probably just going to end up inducing cytotoxicity and also have a low transfection efficiency. And then lastly, for me, consistency is very important. Actually, I think that if you have a little bit of OCD, this is always a good thing when it comes to transfections. Making sure you always see the exact same amount of cells. They are showing the exact same level of confluency. You're mixing everything exactly the same. It goes a very long way in making sure your experiments go the way they should. I mean, there's so many ways where so many different points of variation can come into your experiment. So, and for you to be able to avoid variation from your transfection step will help a lot in the end with getting a successful experiment. And so lastly, I just thought I'd share this somewhere where I actually learned a lot from working with plasmids is a website called AdGene. It's actually a non-profit organization where they collect plasmids from researchers all around the world, and then they share them, and you can actually buy it, and it's available to buy from anyone. And what they have a large resource available about telling you everything you need to know about working with plasmids, from making them to transfecting them, and then have an entire section dedicated to fluorescent proteins and fluorescent protein plasmids. So say you're interested in looking for plasmid that where you can create a tag to your protein of interest, or labeling a cellular compartment, or even if you just need an empty plasmid to help you with optimizing your transfection, they have everything available that you can think of. And because they're non-profit, it works out a lot cheaper than most companies. So it really helps a lot. And yeah, so that's basically what I have to say. And yeah, and if you have any questions, feel free to ask.
Hi, Tanya. Thank you so much.
It's a pleasure.
I actually wanted to know, so I'm planning on doing a siRNA based transfection. And if you go back a few slides, sorry, I actually forgot the question that I had there. But I wanted to know that whether, sorry. Does anyone else have a question so long? While I try to remember what I was going to ask.
I have a question. If you can go back to the slide where you show the fluorescence.
This one.
Yes, that one. I would like to know, and this is maybe a very silly question, but can you control the concentration of the plasmid? So what I'm trying to say, like a lower concentration would mean a lower frequency of transfection would lead to a lower signal, and then just a high frequency of transfection would lead to a higher signal. Can you control the output?
Yes, that is actually one of the things that you would most likely end up optimizing. But it's not as, if you use more plasmid, you will most likely reach a point where you start to stress out the cell.
Okay.
So in this case, more is not always better. For most cases, if you're using a transfection reagent, you can start off using different ratios of, for instance, plasmid DNA versus the transfection reagent. And every cell line would then require a specific ratio. So in some cases, you can have cell lines where you don't have to transfect a lot of DNA and you still get very good signal. Whereas in other cell lines, you might need a little bit more to get a good signal. But in most cases, well, yeah, when it comes to transfections, more doesn't necessarily mean better. Because since you have to keep a ratio between the plasmid DNA and the reagent, if you add more of the plasmid DNA, you'll probably end up adding more of the reagent. And that might stress out the cell, and you can actually, and it can lead to cell death.
Okay. So if you run your samples on a flow cytometer and you get a really, really bright signal, you can maybe use less plasmid to just reduce that signal. Is it making sense what I'm trying to ask?
Well, if the signal from the fluorescence, I mean, in most cases, the signal from the fluorescence will be what you would expect from that fluorescent molecule. But I have seen that sometimes certain cells just don't like, for instance, fluorescent proteins, and certain cell lines are also really quite resistant to transfection. So they might end up moving the plasmid, for instance, into some kind of vesicle, and then which can also lead to degrading the plasmid, which can also then decrease your signal. But in most cases, the signal intensity should be quite good, and it should be the same as you would expect from what is normally expected from that fluorescent molecule.
Oh, okay, thank you. Tariq, did you remember your question?
Yes. Thank you for the time. So in our department, they have a nucleofector, which uses electroporation. And what they've told me is that there's a lot of cell death that results from the electroporation. And also they couldn't, for some reason, they couldn't get, even with help, etc., they couldn't get good enough efficiency with it. So what other groups within the department do is that they just incubate the siRNA with their cells for a certain time, and they get good enough transfection. So that's kind of like passive without any vector. Have you got any experience with that, and is it advisable?
Well, to be honest, I mean, I've used siRNAs before, but for that I've used a transfection reagent, alongside that. To be honest, I don't know how. I mean, you say they get sufficient, efficiency, and that they're seeing an effect. But I would expect that by just exposing the cells to the siRNA, since it's RNA molecules, RNA is notoriously less stable than, for instance, DNA. And the cells can actually express RNases that can degrade RNA molecules. So I'm actually quite surprised that they're actually seeing an effect that's good enough for them. But I think for me, what I would suggest is using it with a transfection reagent so that you know it's a little bit, the delivery to the cells are a little bit more optimized.
Yeah, that makes sense. So I was actually thinking of going and taking control of the nucleofector myself and figuring out what everyone was doing wrong.
Yeah, I know the electroporation, some people are a bit wary about it because you're basically introducing gaps into the membrane. If the cells can't recover fast enough, they will die. But you can play around a lot with the voltages and how many pulses it gives to the cells. I mean, sometimes you can either give 1 or 2 or 3 pulses, and each pulse can last a specific amount of time. So sometimes if you just decrease the number of pulses, technically then you're decreasing the amount of times that your cell membrane is being, attacked. So that could be a little bit less cytotoxic. But yeah, there's a lot of things I know that you can play around with that.
All right. And then so one thing that we wanted to do is try to knock down a gene in patient cells, in PBMCs. How does that differ from cultured cells? And would I need to consider different things?
It is actually, you technically don't need to consider anything else. But I do know that primary cells are notoriously difficult to transfect. So in this case, if you're using electroporation, I would try to find if they already have, if the company that, the brand of the machine that you have, if they maybe have a optimized protocol already specifically for PBMCs. It's quite well known, even with lipid-based transfection reagents, primary cells are more difficult to transfect. So if you can find, someone who has done it before, specifically with PBMCs, I think that could help you quite a bit. But otherwise, the process is exactly the same, yeah.
All right, thanks.
Okay, that seems to be the end of our session. Thank you very much, Tanya, for your time and for the presentation, for setting up the presentation, and for your time telling us more about transfection.
Yeah, it's a pleasure. Thank you, everyone, for listening.
Thank you, everyone. Have a good weekend.
You all too.
Bye.
Bye.
Thanks, Tanya. Bye.